So today we have with us Dr. Bruno Galindo. Dr. Galindo has a bachelor's in biology and a master's and a PhD in genetic and molecular biology. He has been a professor at the State University of the Northern of Parana in Brazil since 2007, where he has been conducting his research in conservation and diversity of neurotropical fishes. He has also contributed to the implementation of sustainability policy at the university. And between 2014 to 2022, he acted as the Dean of Planning at the same university. And currently, he is a visiting professor in CGIL at the University of Guelph. So thank you for joining us, and I'll give the floor over to you.
Okay, Christine, thank you. Let me just put my presentation in full screen mode. Can you see my presentation?
Yes, looks good.
Okay, so thank you, Christine. Thank you, Samla, for the invitation. Thank you for having me this afternoon here at the CGIL Seminar. So today I'm gonna talk a little bit different standpoint about the genetic diversity, but at the end of the day, genetics is genetics. So we can realize many shared points between these two fields. So my presentation this afternoon is entitled Insights of Genetic Diversity Distribution within a Neotropical Watershed. So I am from the State University of the Northern of Paraná, and I am from this laboratory called GECON. It's a conservation genetic laboratory. And I choose these two pictures to start my presentation because here we have two levels of biodiversity, the species level and the molecular level that we are going to go a little bit deeper this afternoon. Just me. Okay, so I bring here two maps. This map from Brazil, South America, and here I have the Paraná state. If we zoom in this part, we can see here the Paraná state, and my university is located in the north of Paraná. We have three campuses, Cornélio Procópio, Bandeirantes, and Jacarezinho. We have about 5,000 students and 500 faculties, just to a little introduction. So this is the outline of our conversation today. I will start giving you a little background about fish diversity, about dams and threats to fish diversity in neotropical region, and a little background about Paranapanema watershed. And then I will present you two cases. One, trying to highlight the role of free flow tributaries, and another trying to highlight the different patterns of distribution of genetic diversity in these watersheds, and some final thoughts. So, biodiversity is a broad concept. We have several levels, several standpoints from where we can see biodiversity. The first one is the biome level. We have different biomes covering the earth. And then we have the environment level. We have different environments within one biome. For example, as I'm talking about fishes, in the aquatic environment, we have the oceans, lake, rivers, streams, so on. And then within each environment, we have different species. So here I brought three species from our research. And within each species, we have different genetic composition. So we are going to focus in this level today, genetic level, so we can assess this level with different molecular markers. So let's look to the freshwater environment. So here I have the world map. We have about 71% of the Earth's surface covered by marine environments. The other 29% are covered by lands, and only 1% of the Earth is covered by surface fresh waters. I mean rivers, lakes, and swamps. So, in the whole world, we have about 36,000 fish species, and they are divided in this way. For example, in this 71% of Earth's coverage, I have about 18,000 species. And in these 1%, I have also 18,000 species. So we can realize the importance of freshwater environment with these numbers. In Brazil, we have about 3,000 species in freshwater species. So this is our study area. Again, Brazil. I have this yellow rectangle here, and I have here the Paraná state. Just to give you a sense of location, I have here the São Paulo state, Rio de Janeiro state, Paraguay, and Argentina. So we are going to focus in this blue ellipse that is the Paranapanema watershed. So this basin has recorded so far 225 species of fishes. And in the neotropical area, but I could say in the whole world, we have several kinds of threats to freshwater fauna. And I could say overexploration, deforestation, pollution, introduction of exotic species, and dam construction. And today I'm gonna focus only in dam construction, and I'm gonna address this topic in a very, very simple way, okay? So, for example, here I have a river in a pre-impoundment state. So we have different habitats. We have calm waters, we have rapid waters, we have small space, big space. We have different levels of water. So we have water level fluctuation, different between drought periods and flooding periods. So we have a very, very active environment. But when a dam is built into a river, several consequences arise. For example, the dam itself will block migratory routes. It's pretty obvious. Will control the water flow, so that water flow, that difference in water flow, maybe disappear with the dam, and a pretty direct consequences that the dam dividing the population, so will diminish the effective number. Another consequence is the elimination of breeding and spawning areas by the lake formation. And another consequence is the dam and the lake will change several water parameters. For example, pH, oxygen availability, temperature, so a lot of difference will arise. My slide's not going. Okay, yeah. So a little bit slow. Let me just try something different here. Again. Oh, I'm sorry, I don't know what's happening. Okay, maybe now. Okay, so in Brazil, primarily, we have two mitigating measures to cope with the river impoundment. The first one are transposition device like this fish ladder, and the second one is fish stocking that consists in raising fishes and releasing them into the rivers. So I was I'm going to start talking a little bit about some issues with fish stocking. For example, usually they use a small number of species in these programs. For example, I told you in the Paranapanema watershed we have about 225 species, but I can't imagine one stocking program with more than 10 species. Usually they use 6, 5, but yes, let's consider 10 is still a low number, a pretty low number comparing with the total number of species. And usually the focus is on species with a commercial value, and usually they have a small size of breeding stock, and sometimes they use animals from other watersheds as breeding stock. So what we can see here on the left side, we have one species that has been used in fish stocking programs. And on the right side, we have one haplotype network. These are haplotypes network from mitochondrial DNA. In the right side, we have one species that has not been used in fish stocking program. What we can see here is in the left side, we can't identify any pattern, any logical evolution because we have a mass. In this situation, they have used uh, species, animals from other watersheds as breed stock. So this could have a harmful consequence for the fish stock. Another mitigatory measure is to build a transposition device. When we have a transposition device as a fish ladder, you can see, let me pick the pointer. You can see over here. Uh, Usually, these devices are more attractive than the tributary. So fishes usually will prefer go into this device. And when the fish get upstream areas, they won't find any proper area for reproduction. So they want reproduction. And they can't find the way back to the downstream environment. So on the other hand, if we didn't have this transposition device, fish could probably find this tributary and reproduce into this tributary. So I'm gonna talk a little bit about dams in Paranapanema watershed. So each dot, regardless of the color, is one hydropower plant. So if you remember, this blue ellipse is our study area. So we have a lot hydropower plants in our study area. As a matter of fact, Brazil is changing its electric matrix in the last years. As you can see, one increment in solar generation and wind generation, and a lower dependency on hydroelectricity. But Brazil is still increasing the number of hydroelectric power plants. If you see this picture, we have each dot is one hydro power plant in operation. In this picture, you will see each dot is one hydropower plant under construction. And in this picture, one project to a new hydropower plant construction approved for the Brazilian's government department accountable for this. So Brazil is still increasing the number of hydro power plants. So I'm gonna shift a little bit the focus of the presentation in this point, just to talk briefly about the two kinds of molecular marker we used in our research project that I'm going to present you this afternoon. So the first one is microsatellite, also known as SSR, that stands for single sequence repeat. So this kind of molecular marker are repetitions in tandem of a motif of a unity from 1 to 6 nucleotides, but 2 is the most common. So this animal, I have in this chromosome, I have 1, 2, 3, 4, 5 repetitions, and in the other homologous chromosome, 1, 2, 3, 4, 5 repetitions. So this animal is homozygous. This animal, in the other side, is a heterozygote. This animal has 1, 2, 3, 4 repetitions in one chromosome and 6 repetition on the homologous chromosome. So when we see the outcome, we will see one band for this animal and 2 bands for these animals. And the visualization of the genotype can be done in acrylamide gel, but nowadays we are doing in sequencers. The other molecular marker we used in our project is from mitochondrial DNA and is called D-loop. D-loop is a no-coding portion of the control region of the mitochondrial DNA. So it's has a different kind of inheritance and a different evolution rate of evolution. So now I'm going to present you the first case. The first case, this paper was carried out, this research was carried out by Diego during his PhD in collaboration with Dr. Sofia from another university in our state, the State University of Londrina. And in this research project, we were interested in analyze the genetic relationship between reservoirs and its free flow tributaries. So to carry out this job, we needed to find a species that was broadly distributed through the watershed and that had not been used in fish stock program because we wouldn't interested in any human interference in our results. So this is the Paranapanema watershed, and this is the Paranapanema River. Each blue triangle here is one hydropower plant in the main channel, and each yellow triangle here is one hydropower plant in one tributary. But we still have some free flow tributary, or at least tributary with free flow strats. As for example, Pirapó River, as for example, Congonhas River, and so on. So we performed population structure analysis using 3 different hierarchical levels. Our first comparison was among all collection sites, so each collection site, and we had a collection site in Paraná River. Paraná River is the river where Itaipu hydropower plant is built. Itaipu was the biggest hydropower plant in the world, and now placed the second is in the second place. So the second hierarchical level, we grouped each reservoir with its tributary. So here I have Rosana Reservoir and its tributary, Capivara Reservoir and its tributaries, Chavantes Reservoir and its tributaries, and Jurumirim Reservoir and its tributaries. And the third hierarchical cluster was, we grouped all collection sites downstream the Salto Grande Falls, that is a natural barrier, and all collection sites upstream Salto Grande Falls. So the first outcome I would like to share with you is this. We have here some results, and as we can see in this table, regardless the group formation 1, 2, or 3, the primary source of variation was found within population. And here I would like to compare each reservoir against its tributaries. Here I have microsatellite data, here I have mitochondrial data, and I'm looking for population structure. As I know the numbers are pretty small in this table, I highlighted with blue non-significant results, and I highlighted with orange, this one, when the outcome was significant, but the value of genetic structuration was pretty low, okay? So as we can see here, in general, here I highlighted all comparison between reservoir and river, and the reservoir and its tributaries. And as we can see, we have a predominance of low or non-significant values, which means that we have between reservoirs and its tributaries, the genetic diversity is pretty homogeneous distributed. On the other side, when we compare different reservoirs separated by a dam, so I have one reservoir, a dam, and another reservoir, when we made this comparison, we found only the predominance of significant values, significant structure, genetic structure values, which means that between reservoirs separated by dams, we have a predominance of significant genetic substructuring. So let's take a look in this attribution. What we can see is that Paraná River is pretty different. So Paraná River, where we have the Itaipu Dam, is for sure another population. And here we have the Paranapanema River. What we can realize here in this yellow dotted line is the Salto Grande Falls, that natural barrier I told you. So Salto Grande Falls apparently is starting a differentiation trending between downstream sites and upstream sites. And despite the AMOV analysis, it didn't show a huge amount of variation between upstream and downstream Salto Grande Falls. We can observe in this slide that, so if we look this group, upstream group, we have 5 mitochondrial haplotypes and 33 microsatellite alleles that we found in upstream, but we don't found downstream. And if you look this downstream group, we found 106 microsatellite alleles that we didn't found upstream. So this is a pretty strong evidence that we have at least the beginning of 2 groups. So in summary, this first case, I would like to stress the use of free-flowing threads of tributary has allowed the genetic flow among different areas. And the maintenance of large effective population size and consequently satisfactory genetic diversity levels. And we could also say that different stock using the same free flow tributary could also be a factor, minimizing pre-existent populations difference during subsequent generation. Now, I'm going to start to present you the second case I choose for this afternoon. In this second case, was carried out by Carolina Polinario during her PhD and also in collaboration with Dr. Sofia from the State University of Londrina. And in this research project, our goal was to highlight the importance of streams to preserve the genetic diversity. Again, we needed a species that was evenly distributed through the watershed, but this species should occupy as well streams, not only main channels of rivers. And this species is Hippopotamus ancestroides, also known as cascudo. But what motivated this project was one interesting finding in previous project. I guess was during the monography of done by Diego. I am not sure, but one previous project show us that sometimes in a stream we have more structured population between collection sites apart 4 kilometers than in a river between collection sites 40 kilometers apart. So sometimes we have more genetic differentiation between stream than between rivers. So we would like to understand this distribution of genetic diversity through the main channel and its streams, its small tributaries, its streams. So we pick Laranjinha River here, Laranjinha River, the main channel, these red dots. We choose 7 collection sites and we choose 6 streams, 2 streams in the upper part, 2 streams in the middle part, and 2 streams in the lower part. And each streams we collect in the upper, middle, and lower part. So this was our study area. And here we have some results. And as I told you, I would like to highlight the importance of streams for genetic conservation in fresh waters. So I start highlighting. So here I have the 7 collection sites in the river, and here all collection sites in streams, and you can see the number of private alleles we found in streams. We also found 4 haplotypes restricted to streams. So here I have the observed homozygous number ranging from 0.5 to 0.7. The heterozygosity, expected heterozygosity ranging from 0.4 to 0.7. And these values are pretty decent values for neotropical fishes. And maybe because these we didn't found much inbreeding as significant inbreeding coefficient, only this string presented some significant value. When we start to look to mitochondrial outcomes, we realize that usually the diversity index are below 0.5, but we still have numbers as high as 0.8, and the nucleotide diversity is pretty low. But what I would like to highlight for you in this slide is these 2 last columns, the Tajima and FAS neutrality test. We found a lot of negative and significant value, which can be one hint of population expansion. So, and we also found an interesting pattern of longitudinal distribution of genetic diversity. So this is the water flow in the river and in a stream. And what we found was that the genetic diversity increased towards the mouth. So, and why? Why this genetic diversity increased towards the mouth? So I would like to say that the same pattern is valid for another level of biodiversity, the species level. So here close to the mouth, I have more species than here close to the source. So the same increasing in diversity is valid for species level and genetic level. But why? Maybe because these areas are larger sites than here, and larger areas uh, can have more capacity to sustain larger populations. So here I have a bigger number of individuals than here. And if I have a bigger number of individuals, I have less action of genetic drift. So here I have a small number of individuals, so I have a big action of genetic drift. And as you know, genetic drift can eliminate genetic diversity as well. Another reason why we could have more diversity close to the mouth than close to the source is that usually the mouth is closer and is more susceptible to receive migrants from other genetic units. So the genetic diversity, as I told you, we found this is a haplotype network. Here we have each circle is one haplotype. The bigger the circle, the bigger the number of individuals. And in these arrows, I have some haplotypes that are restricted to strings. So these haplotypes are not found in the main channel, only in strings. And here, I would like to highlight this group of haplotypes. So this group of haplotypes is another mitochondrial lineage. And we can say this because we have a lot of mutational steps between this lineage and this lineage. And we didn't find any intermediary haplotype. So for sure, this is another lineage. So this is one area of secondary contact between two lineages of mitochondrial haplotypes. And we found, as I told you, higher mitochondrial diversity and higher microsatellite diversity as well, but now I'm going to focus in mitochondria. We have higher mitochondrial diversity in the lower areas than in the upper areas. And what this means is that maybe we can infer that the movement of colonization of this basin was in this direction from the source, from the mouth to the source. So because at the beginning, this species has occupied the lower areas and then moved through the upper areas. So all our demographic analysis of mitochondrial DNA indicated population expansion. Our haplotype network has a star shape. Oh, I'm sorry, I forgot to say this. This is a star shape when we have one main haplotype with that a lot of individuals has this haplotype, and we have brands from these haplotypes with small number of mutations. So this is our star shape, and this also indicates a population expansion. So our haplotype network, our D and FS test, and our mismatch distribution, all these parameters indicated a population expansion. So our species are expanding its area of occurrence. And this is also what I was showing here. So probably our species is expanding its occupational area. So we also have some data of recent gene flow, and we found that the main part of the population are composed by non-migrant individuals. And I would like to show you again this attribution analysis. I have here a pretty interesting graph with k equal 2, or which means I had two ancient population forming this group of fishes, group of individuals. So this red population are the lower part of the river and its streams, and the green part is the middle and higher part of our basin. But we can go further and rerun the attribution analysis just using these individuals and separating these individuals. So we rerun two different analyses. For this group of individuals, we also found a k equal 2, and for this group of individuals, we found a k equal 3. We can see here that we have pretty distinct group of individuals as this collection site and as this stream. But in general, we can see that our river, our basin, is divided into upper part, middle part, and lower part. So, but I told you at the beginning of this presentation that what motivated this research was one interesting finding that we had more genetic structure into a stream than into the river. And in this research, the average distance between two collection sites in the river was 37.9 meters, and the average distance between two collection sites in the stream was 8.9 kilometers. But when we look the population structure, the average population structure into the stream, we found always bigger values than along the river. But why this could happen? The first reason could be related why streams has more genetic structure, more genetic differentiation than rivers. The first reason could be because streams are less stable environments. They have less volume, so they are more prone. They are more prone to temperature oscillations, pollutants, drought periods. So these are less stable environments. And another reason could be related to the biology of these species, because our results suggest a limited displacement of Epostomus ancestroids. The genetic differentiation we found between short or long distance, we found correlation between geographical distance and genetic differentiation, and we found some haplotypes limited to only one stream. So all this evidence reinforce this aspect of the biology of this species, that this is a species with limited displacement. And limited displacement is fit pretty well with stepstone model of migration of gene flow. Our results, the stream features, and the biology of the species fits pretty well with stepstone model. What's the stepstone model? Stepstone's model is a gene flow model where each collection site, each population will change alleles only with the next population. So this population only change alleles with these only with these, the site C only with sites D and B, and so on. So this model of gene flow is pretty fits pretty well with our finds. So I'm almost reaching the end. So what I would like to say with this, what I would like to show with this, the last case I show you is that we have a genetic makeup in a river or in a basement or in a basin or in a environment, and we have stream dynamics and aspects of the biology of the species aging over this genetic makeup over the time. And this leads to a differentiation between, so I will pick some part of this genetic makeup and another part and another part. So I will start to have private alleles in this section, another private allele in this section, different allele frequencies in this section. So I will start to differentiate this original genetic makeup with these dynamics, some aspects of biology of this species and some dynamics of the stream. They are less stable environments. And we can measure these differences with genetic diversity estimates like mitochondrial DNA or microsatellite, haplotypes network, genetic differentiation, levels and structure results. So all of these can help, all of these tools can help us to understand this differentiation process. But related to the conservation scenario, what I would like to highlight is that, let's say that this collection site has a lot of private alleles, a lot of private haplotypes, different proportion of alleles. So if something happens, for example, pollution or deforestation, and if this population disappear, all this genetic variation will disappear. So what I mean is that the genetic diversity, and when I say genetic diversity, I'm also saying evolutionary potential is heterogeneous distributed through the watershed for some species. And tributaries, regardless a small stream or a big rivers, have important role in the maintenance of genetic diversity in the watershed. And also that is important to use different molecular markers because each one capture different aspects of genetic composition and evolutionary history. So this was what I had to this afternoon. I would like to thank the State University of the Northern of Paraná for all the infrastructure, all the logistics this university provided to us. I would like to thank the Araucaria Foundation, is a provincial or state funding agents, and CNPq is also a national funding agents. And I would like to thank another university in our state, the State University of Londrina, where we have important collaboration, special with Professor Sofia. I would like to thank Diego, Caroline, and Wilson that was pretty related with these two papers I presented to you this afternoon, but we have a big group of people that was pretty important to help us to have these outcomes. So thank you again for having me, and I'm here if you have, I'm available if you have any question.
Thank you, Dr. Galindo. We will now open the floor for questions. If anyone has any questions or comments, discussion, feel free to raise your hand and then you can turn on your microphones. Or if you want, you can put your question into the chat. I can read it out loud as well, if you prefer. Flavio?
Yes, I have a question. Can you hear me?
Yes.
Yes. Okay. Yeah. Uh, Bruno, thank you so much for this presentation. It's really very, very interesting. So something outside of things that you do every day here at CGIL, but quite interesting. So I have some um, practical questions for you. So based on the two cases that you presented, especially the first one, so what would be the practical recommendations, when you are building all these new hydropower powers or hydropower stations, right? So how what are the recommendations to try to preserve the diversity, right? That would not cause less damage, let's say. Do you have any recommendations, or your group had the opportunity to recommend what to
Yes,
do
yes.
to minimize, to mitigate the problem?
Mhm. Excellent question, Professor Flavio. Uh, Actually, as I showed in the background part, the two main mitigating measures are not working well. So you can't replace the genetic diversity releasing fishes into the river because usually they use a small number of breed stock, sometimes breed stock from another watershed, and so this measure is not working well. The transposition device are not working well because sometimes they are selective, they are hotspots for predation, they are one-way movement, so fishes can get upstream but can't come back. And when the fish get upstream, fish can't find usually proper environments for reproduction. So these two measures are not working. So our recommendation and the goal of this research project was to prove the importance of free flow tributaries. So you need to leave some free flow tributaries, or at least tributaries with free flow strats. So you can't build one new hydropower plant in each tributary because we are showing and we have a lot of evidence. There is one interesting project from well that they collected larvae and eggs, and they did the barcode to identify the species. So they also proved that some species are using tributaries for reproduction. So our advice is do not build hydropower plants in each tributary. If you have hydropower plants uh, built on the main channel, leave the tributaries free because the fish, the population needs these tributaries to keep reproducing.
Mhm. May I have a second question here that's
Yes,
related? So you showed clear to us that the streams are very important for the diversity,
yes.
right? But
Hmm.
the problem with this small, these streams, they are located within private properties completely, right?
Yes.
Because they are small, right?
Hmm.
So then it depends on the private owners to keep that stream healthy, right? No
Hmm.
pollution, not keeping vegetation around and so on. So that's a dilemma, right? Because they are the main source of diversity, but at the same time, the public agencies have a very hard time to interfere, right? Because a big river, there's no owner, let's say, not
Hmm.
owner between quotes, but a stream is really private within private properties. So I think it's really, really just education, right? There is no other way.
Yes, yes. Actually, while a river can reach far 400 kilometers, 400 kilometers. I stream sometimes 29, 28 kilometers. So one or two properties the stream can be into one or two properties. Actually, Brazil has legislation. The water bodies do not belong to people, belong to the the country, and we have I don't know how to say in English, but we have riparian vegetation that should be maintained by the owners. The owners need to maintain these forests along the streams, and this is the point. This is the point because it's hard to keep
To enforce.
the to enforce. Yes,
Yeah.
and should be very important because and also for terrestrial animals because this could form genetic hallways. I don't know how to say in English. I'm sorry, but the terrestrial animals could use these riparian forests to make
To
movement
move.
between areas.
Yeah. My last one, very quick. This is more I like to learn. So this mitochondrial DNA, the D loop part that you are
Yes.
using, is the same one that is used for describing the species, to identify different species, or is it a different part of the mitochondrial
Yes,
DNA?
it's a different part.
Yeah. Okay.
The part the we use this part of the orange of replication is called D loop, and to perform the DNA barcode, usually they use one part of cytochrome oxidase one. So this sequence has about 400 base pairs, and the barcode, the cytochrome oxidase one, has about 800. I'm sorry, 800. So it's a different sequence. I'm not sure, but I guess the barcode evolve faster than the loop.
Okay. Okay. Thanks, Bruno. Thanks.
Thank you, Professor.
Salma?
Hi, Bruno. Thank you for your presentation. It was amazing.
Hi, Samla.
Hi. It's nice to also
Thank
hear
you.
about different stuff that we normally work with. But I was wondering, I saw that you guys use microsatellites, and of course, there's a different application when you read it in conservation. But I was wondering if it is possible to use like SNP markers, especially the ones that has more density. And
Mhm.
if it is used or if it is not used, why is not used?
Mhm. Excellent question. Actually, we use some SNPs when we are talking about mitochondrial DNA, but is not in comparing with SNP panels. We are talking about, I don't know, 24, 25 SNPs in a mitochondrial DNA when in the loop, especially when the SNP panel are way more big. But the question is, the SNP panels usually they are developed to commercial species. So, for example, I know that we have SNP panels for salmon, for tilapia, and maybe for other species. What we don't have available for uh, native species so far. So if we decided, as we have some species pretty popular in Brazil and pretty spread through the almost whole country, and maybe if someday one could develop some Z panels, we could start work with. But at my knowledge, we do not have so far. But would be pretty interesting. We would have much more power, especially in admixture analysis, and I have seen some interesting paper with population genetics, phylogeography using SNP panels with commercial breeds as goats, sheep, and even for humans that are pretty interesting.
Oh, nice. Yeah, thank you. So it is possible to use, but the problem is that we don't have the panels available.
Yes, exactly, exactly. I didn't mention, but we have a lot of wet work with this, this to do this. A lot of PCR, a lot of DNA extraction, a lot of work to catch the fishes. So it's a lot of wet work.
Yeah, I can imagine how many gels you'd have to do.
Yeah, yeah.
Okay, thank you.
Thank you again.
Okay, do we
It's
have
lit,
any other?
and it's just a joke. It's literally a wet lab
Yeah.
working fish.
Literally and wet lab.
So if there is no other question, I have another question.
Sure, Flavio, go
So
ahead.
I wonder. So you showed us that close to the mouth of the river, right?
Mhm.
You have a broader diversity of species,
Yes.
right? Because there is maybe more space and so on, as you mentioned. But
Mhm.
I wonder if this, when the river is the mouth is by the sea, so then you have
Mhm.
like salt water and fresh water. Do you know if you have a more diversity in that case when the river is flowing towards the sea?
No, I don't know, Professor, but I can imagine that could be a pretty rich environment because usually we have swamps in that area, and that swamps are areas for reproduction of seawater species and also for freshwater species. So I don't know the answer for this question, but I can imagine that this is a pretty rich environment as well.
Yeah. No, I was just thinking in the within the river, not the swamp,
Ah, okay, okay.
because of
No,
the change of the water. Like depend on the level of the sea, then the salt water is going to flow into the river, and
yes,
then as
exactly.
parts of the day will be the opposite. So so how that fluctuation would affect the richness of the species.
E- Exactly. Should for sure we have some guys working with this in Brazil or here in Canada and the US, and I could look this some paper to check because probably could be a pretty interesting outcome.
Yeah, thanks again, Bruno. Very
Thank you,
interesting topic.
thank you, Professor.
We have probably time for one more question if anyone in the audience has another question. I was wondering, what kind of genetic indicators would tell you that a population diversity is becoming kind of too low,
Mhm.
to maybe need some intervention? Or is there any intervention that any intervention that could be done to preserve or improve the diversity of the population?
Yes, we have a lot of indicators, and usually we compare with literature. So we can compare with closed groups, the levels that other researchers have recorded. So, for example, we got for Hippopotamus ancestroides 0.7 expected heterozygosity is a pretty good level of heterozygosity. So I would say that zero point 0.2, below zero point 0.5, we could be careful. Unfortunately, because the time I didn't present another paper that we found a threatened species, one endangered species called Briconatereri. She This species is in the Red Book of Brazilian fauna, and we found a pretty decent level of genetic diversity for microsatellite. Zero point 0.6 of expected heterozygosity is pretty good. But when we looked to the mitochondrial diversity, we found a pretty low nucleotide diversity, pretty low. And this low nucleotide diversity indicated to us an ancient bottleneck in this population. And the microsatellite didn't catch this bottleneck because microsatellite is proper to recent bottleneck, not ancient bottleneck. So our conclusion for this population, because we measure the heterozygosity diversity for microsatellite, we measured the Hardenberg equilibrium, we measured the inbreeding coefficient. So for this population, good microsatellite diversity, no inbreeding, no deviation for Hardenberg equilibrium. But we had low diversity for mitochondria. So our thought is that this population suffer a bottleneck ancient, but is recovering as the microsatellites show us. So the topic for this population, our recommendation was, in our paper was, we don't need to do nothing with this population. The unique thing we need to do is keep monitoring this population to if in the future we need another intervention. And another intervention could be bring some animals from other population, but population which doesn't have a huge genetic difference within this population. But so we can look for another parameters, and because this I told you is pretty important to use different molecular markers to catch different times and different evolutional levels. So and these populations that I showed you are pretty genetic, has a good genetic background.
Yeah, yeah, interesting. Interesting that they can recover by themselves too. Okay, we do. Maybe we could quickly answer this one last question in the chat. Can we rely only on mitochondrial DNA as it only covers the maternal side of genetic information, or do we need to perform genomic DNA, use diversity estimation alone with D-loop or other mitochondrial DNA analysis to study for a proper estimation of diversity parameters in a population?
Oh, let me check the
Yeah.
question here. Yes, so we can usually the D-loop the mitochondrial DNA, they have a small effective number than microsatellite. So they will be pretty susceptible to bottlenecks, and they will show us a history from more ancient bottlenecks than from recent bottlenecks. So I would not perform any populational studies only with mitochondrial DNA, because that if I was only looking for the mitochondrial DNA in this example I told you, maybe I could advise someone to increment this population with new individuals, because we were looking at pretty low nucleotide diversity mainly. But when we look to the microsatellite, that is a molecular marker that evolved faster than the mitochondrial DNA, I realized that no, this population, yes, she suffered a bottleneck in the past, but she's recovering. So I guess the D-loop, the mitochondrial DNA, will tell us one part of the history only, and usually a more ancient part of the history, not a more recent part. So I would not rely, but I would not use only mitochondrial DNA to perform any populational studies. And when you are working with phylogenies, for example, evolutionary studies, you also will try to incorporate some nuclear sequences and not only mitochondrial sequence in your job. I'm not sure if I answered the question properly. Okay.
Okay, great. Thank you. Thank you, Dr. Galindo, and thank you everyone who joined us today. It was a very nice presentation, and it's interesting to learn about natural populations. So it was great. So thank you again, and see you all next week, and have a nice weekend.
Thank you. My pleasure.